and we're streaming live from the University of California, San Francisco at the Weill Institute for Neurosciences, which is an umbrella organization that unites all of the neurosciences at the University of California, San Francisco. The basic sciences, the engineering and the data sciences, along with the clinical disease neurosciences communities. And we are honored, I'm certainly honored, for UCSF and the Weill Institute to be one of the institutions selected for this morning's live stream. And as you know, this is one of a group of workshops that will also be streamed from different institutions around the world to a very broad audience of academicians, industry partners, and the general public. And the goal is to speak about the excitement of neuroscience, the incredible potential that current and future discoveries will have to make all our lives better. Excellent. So this is one of 13 international neuroscience institutes that have been selected to participate in this global event. You can see the institutes here. We're again honored to be one of the selected groups. And special thanks to Professor Comey for his massive undertaking of organizing this global neuroscience initiative. This morning's seminar will speak about one of the treatable neurodegenerative diseases, multiple sclerosis. And in keeping with the theme of neurodegeneration across the conference, we will be focusing on 5 different areas of monitoring neurodegeneration in multiple sclerosis, which is an immune-mediated neurodegenerative disease. First, we will look into advances in visualizing neurodegeneration in MS, and Dr. Riley Bove will speak about this important subject. Second, how can we remotely monitor our patients? And Valerie Block will be our second speaker. Third, we all know that the MRI and modern neuroimaging techniques have and are revolutionizing our ability to detect neurodegeneration. And Dr. Roland Henry, who will be speaking with us remotely, will tell us about advances in this important area. Another enormous opportunity are the new platforms for parallel screening of antibodies in the immune system. This has great opportunity not only to detect signals of the body's response to damage, but also potentially identify pathways for therapy, for treatment, prevention, and repair. Dr. Wilson, Michael Wilson, will speak about that. And last but not least will be Dr. Sergio Baranzini, who will be speaking to us about the very exciting, revolutionary advances in our understanding of the microbiome around us and in us, and how these advances in microbiome biology can help us monitor as well as understand and possibly even treat neurodegeneration. So I think this is going to be an exciting and enjoyable streaming event. After the 5 brief presentations, we will have a roundtable discussion. So with that as an introduction, and again, with thanks to all of you from around the world who are joining us here at UCSF, Dr. Bove will be our first speaker, visualizing neurodegeneration.
All right, well, hello, everybody. Thank you for the kind introduction, Dr. Hauser. It's a pleasure to see you all, or not see you all virtually. I'll be speaking about how to visualize neurodegeneration in the clinic. And when we think about what is the actual goal of precision medicine, what is the actual vision? The vision is to bring all these advanced computational tools, the patient's perspective, iterative data science, really to bear on a specific patient in the clinic. And how do we actually make this work? You're going to hear from my colleagues who are going to present beautiful data on neuroimaging, on wearables, on fancy omics that enable us to really see treatment response or molecular signatures at a very, very detailed level. And then we can think about what actually happens when we're in the clinic. And in the clinic, we really have much more basic clinical tools. As neurologists, we have a reflex hammer. We sort of have a clunky MRI review system. We don't really have a way to bring all these fancy pieces of data science and sort of augmented information about patients to bear in the clinic. And really, to make precision medicine work, you have to deliver information using the 5 right principle, the right information for treatment to the right person in the right format, through the right channel, and at the right time in the clinical workflow for it to actually be implemented and effective in the clinical setting. And that's the focus of our Bridge Precision Medicine platform. And that's what I'll be speaking with you about. And so what is Bridge? On a very, very simple level, it's a website, and it displays information about a given patient. And it launches from our EHR, electronic health record, electronic medical record at UCSF. That's the EPIC system. And it launches with one click button. And it consists of different widgets, which is sort of visualizations that pull information from different sources. And Bridge dashboards are really customized for the clinical needs of a given clinic. And so the idea is to place the clinical data alongside the research data and computational resources so that you can have an integrated approach. And as you can see here, these are just some examples of data sources. Bridge can pull data from the EHR directly through, or we can also do advanced sort of computation on the data so that they can be processed in new ways and then visualized. And the bridge is really designed using a modular design system so that we can expand to other clinics, other dashboards in a fairly efficient way and streamlined approach. And so now live at UCSF, we have a number of dashboards across the neurosciences, both in neurology and psychiatry. And each is really customized to enable the display of information, delivery of information that's really relevant to specific clinic, whether that's memory and aging, movement disorders, epilepsy, et cetera. So thinking about what kind of research applications Bridge can really support, there is a sort of an early experience in sort of the use and delivery of precision medicine where, as we all know, warfarin dosing is a very complex problem in the clinical world. It's lengthy to get patients on the right dose of warfarin to sort of thin their blood. There's a lot of risky complications if this isn't done appropriately. It's very sort of costly and time intensive. And there was an early promise that genotyping patients could support more rapid titration of warfarin to the right dose. And in fact, when that was actually truly able to be implemented in the clinic, it was seen that that didn't actually really make a difference. And so when we think about sort of delivering precision medicine, we have to think, like, how do we actually know that it'll truly make a difference for our patients when we're visualizing neurodegeneration in different ways? How does this actually help the delivery of care? And so this is a sort of sort of schematic view of the types of in silico trials that can be supported by a platform like Bridge. You can see the display of information longitudinally over time in the top left widget, and that sort of asks, can we get a more rapid, more comprehensive perspective, and can we really situate that patient's clinical course in the clinical course of other patients with sort of same disease duration or MS type? What does delivery of advanced neuroimaging metrics do to sort of support early diagnosis of specific types of neurodegeneration? And how can we also use sort of different algorithms, different predictive tools more effectively in the clinic? So just this is from a sort of schematic perspective to actually talking about what we're doing a little bit in multiple sclerosis. And I'll talk to you about a couple new sort of data sources that we're bringing in. So the first is you're going to hear from Dr. Henry about sort of the utility of advanced imaging, spinal cord imaging, to really identify neurodegeneration. And so being able to pull in the clinical MRI scans and then pull them through the system and visualize these scans over time so that we can scroll over time and see that a patient's spinal cord is experiencing atrophy, for instance, is one clinical use case. Another is the Spoke knowledge network that Dr. Baranzini has developed and we'll touch on as well. And through information in the EHR, Dr. Baranzini and his team are able to then pull that information through a complex knowledge network, some knowledge magic, and spit out different types of pathways that may be biologically relevant or therapeutically relevant. And so one goal of our integration of Spoke into Bridge is to support us being able to identify which patients may be at risk for specific conditions, what might be a differential diagnosis for a patient presenting with a specific phenotype or syndrome or even problem, and then also how to tailor treatment response. And so this is just an elegant visual from Dr. Baranzini's team, just showing how we may be on a patient-specific level able to identify molecular signatures of progression and then target medications appropriately. Another really exciting area is sort of wearables and digital phenotyping. You'll hear from Dr. Block, who's going to talk about her extensive research in this arena with ambulatory monitoring. But typically patients wear their Fitbits and have all of those data on their apps. And as clinicians, we don't have access to that information in a simple, streamlined way that allows us to do something about that information. And so whether we're talking about wearables worn on the wrist or elsewhere, or smartphone apps, this is a depiction of the Roche Genentech Floodlight app, but there are many, many of these developed. We're now working to bring those data into Bridge so that we can view the data longitudinally and interpret the data in the context of a patient's condition, and also do iterative data science on those data. In the clinic, we have developed digital phenotyping sort of corridor where we're able to phenotype gait, phenotype cognition, phenotype other domains of neurological function, and then both be able to view those data through videos that pull right into the patient's encounter, and also, again, provide a sort of iterative data science on those digital phenotype data, with the goal of being able to more precisely identify how patient function is changing over the long term and what to do about it. And finally, we talk a lot about precision medicine as sort of being a sort of a constellation of omics, but really the goal is also to pull in the patient's voice. Our patients tell us that they are progressing in multiple sclerosis long before we can see it on a lot of clinical metrics. And so being able to actually integrate the patient's voice into the clinical encounter in a more streamlined and efficient way is also a big goal of precision medicine and a big goal of our project. And so just to give you a little sense of a pilot project that we did with Bridge, we did a pilot project where we asked patients to complete a set of validated patient-reported outcomes before the clinic. And then we were able to pull those data right into the Bridge dashboard and visualize those data at the point of care. And what we found were for the clinicians who had been asked to use Bridge and who were able to visualize their patients' mood scores directly at the point of care, there was a significant and substantial increase in attention paid to depression as a sort of decision-making action point during the visit. And so the numbers were slightly enormous and shocking, but the data from this pilot then enabled us to obtain a Department of Defense funded research grant in order to actually expand this work and figuring out how we can remotely capture patient-reported mood and then pull that into the clinical encounter in ways that matter. So in streamlined in-basket messaging for clinicians so they can be alerted and respond in due time, and then also in more built out versions of Bridge where we can have clinical decision support around depression treatment, and including geolocation factors, geocoding factors, such as the availability of resources in a patient's given community to treat their mood or other symptoms. And so to sort of bring this all together, the goal of Bridge is really to be able to pull all of the advanced work that's happening in the research realm and to figure out what of that information pertaining to neurodegeneration is actually actionable for a given patient in a given clinical encounter at the point of care. I want to thank you for your attention. I want to thank my colleagues, so our Bridge team, my lab, and then our UCSF Weill Institute for all the support that we have received to develop this platform, and also for all the collaboration that we enjoy across our group so that we can actually support this bench to bedside work. Thank you.
Application that you mentioned is decision support. And it strikes me that this is a wonderful way to democratize knowledge. Two questions in this area, though. First, not all patients have similar cost-benefit analysis conclusions. Some are risk takers, and some prefer to be very conservative. How can you imagine taking individual preferences into the tool? Maybe I'll start with that question.
So for our sort of global audience, the question is, patients have their own preferences regarding risks and benefits of treatments or approaches. And so how do we actually take into account the patient preferences and their risk tolerance when we're thinking about clinical decision support? And I think that's a critical question and point. The patient, of course, the goal of this precision medicine is to actually tailor everything to that given patient, and their preferences are part of this. And of course, we can make all the recommendations we want, but the patient then goes home and lives in their community and and makes what they will of our recommendations. And so I think building in flexibility into the decision support modules is one key way of doing that. And we call these more action prompts rather than decision support, in the sense that the clinician may be prompted to consider physical therapy, prescribe an orthotic, change medications, et cetera. And then that still is a conversation between the patient and the physician. So we try to bring as comprehensive as action prompts as we can. And then the goal then in the encounter is to drill down on what actually makes sense for the patient. And the sort of geocoding and everything else is really intended to support sort of carrying out what they decide to do in their community, because we know that the burden of living with MS is very, very high.
Then I'll ask the second question. Okay. I'm channeling what our audience members are thinking. It is magnificent to be able to see a snapshot of my patient's course over time displayed so easily and beautifully. How can I access this for my patients?
Yeah. So the question is, Dr. Hauser is saying, but it'd be nice to be able to see such a snapshot and access the tool. And the key, I think, the strength and the complication of this platform is that this platform is embedded in our EHR through sort of, and there's a lot of extensive governance around privacy, information technology, risk, et cetera, security, sort of, knowledge, data security, and all of these things. And so the code for Bridge is shareable, and we are delighted to work with investigators and other health systems who would like sort of the code itself. But each health system has to have its own integration with their EHR for it to truly work. And that is the piece where there really needs to be a champion in a given health system who will take the code, take all of our really sort of detailed experience and know-how about how to integrate with the EHR and the health system, and then see it through in their health system. And so we can imagine a number of similar tools applied across health systems, and sort of figure out what works in one could then be used in another, or tested in another.
Thank you very much.
All right, thank you.
Well, thank you very much for inviting me today, and I'm going to speak about that's a bit like the mute on Zoom. So I was saying thank you very much for inviting me to speak today, and I'm going to speak on remote monitoring as a potential signal for MS progression. These are my disclosures. So why remote monitoring? How often do we as clinicians get to see patients? Neurologists get to see their patients once, twice, maybe 3 times a year. As a physical therapist, I get to see patients for a more concentrated amount of time, but then maybe not again for years, depending on their insurance. So these sporadic measures really don't give us a complete picture of a patient's function or dysfunction. And we miss much of the important variability that underlies potential progression. Remote monitoring, on the other hand, can be collected in the home setting for long periods of time and has the benefit of showing us a broader, better picture of how patients are doing in their ecological setting. So ambulatory impairment is one of the most prevalent consequences And more granular disability metrics are needed to advance research in MS in this area. The current metrics that we have fail to capture the important variability in ambulatory behavior, as many of these outcome measures are carried out in controlled contexts of the clinic, using performance tests that lack this ecological validity. So they're just snapshots of function at a specific point in time. And one of the most commonly used of these is the Expanded Disability Status Scale, so the EDSS. It's highly dependent on ambulatory capacity, restricting its ability to capture differences in physical activity at each level. So an example of this is an EDSS 6, where someone needs a cane or unilateral assistance to walk. And here, we can have someone that is very active and mobile within their community and walks so many miles a day, or someone that's essentially homebound because of their need for a cane. And yet both of these are classified as an EDSS of 6. We also have patient-reported measures, which are, as Dr. Bove was saying, really important to tell us how patients are doing and how they feel that MS symptoms are affecting them. But these suffer from recall bias and unevenly perceived deficits depending on when they're completed. And there are often discrepancies between these and our clinic measures. So then more recently, we have remote accelerometry, which provides granular, continuous information about, in this case, if we're doing remote activity monitoring, about physical activity in the real world. And so for this reason, we set up the FITRIM study, where we enrolled adults with either progressive or relapsing MS that were able to walk for at least 2 minutes with or without an assistive device. And we used the Fitbit Flex to continuously monitor their physical activity over a year. And here the blue arrows indicate time points where performance-based measures were evaluated in the clinics, so the EDSS, a time to 25-foot walk, measuring walking speed, etc. And then the green arrows indicate time points where patient-reported outcomes were completed. And these looked at MS-related symptom impact and how MS subjectively affects walking. And so what we saw here in the graph, you can see on the x-axis, groups of patients by minimal to moderate and increased walking impairment, and on the y-axis, you have the average daily step count averaged over the first month of the study. And what you see is what you'd expect, increased disability shows less physical activity. However, what's notable here is the variance around each EDSS score. So a patient taking 1,000 steps a day is clearly not the same disability as someone taking 7,000 steps a day, and yet these are both scored as an EDSS of 6. When we looked at the data over the year, we saw that patients whose disability score, so their EDSS, improved, trended towards an increase in steps. Those who remained stable in blue showed no significant change in steps, and those who worsened showed a significant decrease in steps here in red. In fact, participants who started out our study doing steps below our cohort median, so around 4,700, had 4 times greater odds of worsening at a year than participants who started the study with a higher level of physical activity. Now, because our cohort was actively treated, most of our patients were in this stable group in blue, so we decided to take a closer look. And here, the dotted line colors represent different EDSS groups within those who remain stable, so no EDSS change. And significant changes in steps were observed in most EDSS groups, from people with low disability at the top there to people with higher disability, EDSS 6 and 6.5 at the bottom. And these results really highlight the use of remote monitoring to capture changes in ambulatory function, even when our clinical disability measures remain stable over the year. So, during the early months of the COVID-19 pandemic, we were really worried about the health and physical activity of our patients, as we know that people with MS are less physically active than healthy controls. And inactivity or sitting is the new smoking. So we collected We collected activity data from participants in one of our own ongoing remote monitoring studies, and there was a statistically, and I think probably more importantly, a clinically meaningful decrease in steps during the week and month after versus before the shelter-in-place order of March 2020, which is shown with the red line there. Now, some individuals adjusted and managed to get back on track. However, overall, after 3 weeks, there were more than 40% that had not recovered to within 10% of their pre-shelter-in-place activity levels. And what we also noted was that people who didn't recover activity started off the study with a lower baseline steps compared to those who rebounded. And this study was actually a really cool way and offered a natural experiment showing that wearables can capture real-time, real-world impact of ecological perturbations and really highlights the way that individuals respond to these and potentially how we can intervene in the future. So remote activity monitoring can also reflect novel neuroimaging measures of disability progression. So as we'll hear from Dr. Henry later on, spinal cord areas correlate with disability and disability progression. Spinal cord atrophy can predict the time to relapse-free disability worsening and progression from relapsing MS to secondary progressive MS. However, the relationship between daily physical activity and spinal cord areas, MS disability, and leg function were unknown. So we looked at quantitative brain and spinal cord imaging and evaluated the association of these with amplitude disability using remote activity monitors over 2 years. And this is a sneak peek of our baseline results that show fewer steps were significantly correlated with smaller C2, C3 gray matter areas and total cord areas, and vice versa. And this provides really exciting preliminary evidence suggesting that spinal cord gray matter area is an anatomical substrate associated with steps. And then steps can therefore serve as a proxy to alert clinicians and researchers about possible changes in structural nervous system pathology. And our longitudinal analysis is ongoing. So what about other functions? Although ambulation and gait dysfunction are some of the most visible and prominent symptoms, there are other more silent symptoms causing just as much disruption to quality of life and even physical activity. And one of these is bladder dysfunction or urinary incontinence. It's a surprisingly common and yet widely understudied symptom. And here again, the evaluation and monitoring of these relies on sporadic questionnaires and snapshots of clinical measures. So we have developed a toolbox with input from clinicians and patient stakeholders over a series of meetings where a range of devices were selected from the many commercially available tools on the market for urinary incontinence and other forms of bladder dysfunction. The tools can monitor bladder change continuously and objectively in real time at home, providing an objective alternative to patient reports of urinary frequency, leakage, and bladder volume. And then these tools and others can be combined with telerehabilitation for a much more closed-loop intervention, increasing the frequency and intensity that we're able to give for bladder interventions. And we're currently enrolling participants and look forward to our preliminary results towards the end of the year. And with that, I'd like to thank all of my mentors, all of the people that were critical in helping me with this data, and happy to take any questions.
Dr. Block. Gesundheit. Monitoring steps seems so much more efficient and inexpensive than the measures that we have. And for our patients, it is so much easier than coming into big academic medical centers and fighting traffic. What do you think, how broadly applicable do you think this will be and should be in the near future? Will most of our patients be using home monitoring sensors such as this?
Well, interestingly, for the the question was how broadly Dr. Hauser was asking if most patients coming into the clinic now will be using remote monitors and how broadly these remote assessments will be going forward. And I want to tell a quick story of the 3 patients that we managed to recruit for the bladder study so far. We're also trying to put Fitbits on them to measure their activity, because once we get into the intervention phase, we want to see if helping with bladder can also help with physical activity. And we have Fitbits for them, and no one wanted our Fitbits because they all have Fitbits already, or they have Apple Watches. So I think it is going to be something that most people have, and they want to have, they want to see, and they were more than happy to share the data with me moving forward.
Fitbit is also monitoring the number of steps.
Yeah.
What about the quality of steps?
That's a very good question. And the Fitbit does a very broad, so if I'm doing this, my Fitbit is telling me that I'm probably doing some steps right now. So it's more physical activity. It's not an exact number of steps. Dr. Bove was talking about the brainwalk, some of the brainwalk initiative, where we're using xenomats, and that gives us a huge amount of data on the quality of steps, so stride width, stride length. The devices, the commercial devices that we have so far don't really give us a good picture of the quality of activity. There are research devices that are worn on the ankle or some of them on the waist that give us really good data, but using those longitudinally is probably going to be tough. But if you wanted to use them for weeks at a time, every month or so, that would probably be.
I think that if I remember correctly, that Apple Watch is now can report out falls. I don't know if that's a capability that it has. It seems like another, again, kind of added metric for patient safety, if that's possible with the Fitbit or.
Right. So the question was, question was that Apple Watches now report falls. So I think they have gyroscopes and accelerometers, and so they can tell what the position of the patient. And that would be great for safety as well. And there are a lot of studies that are using Apple Watches. A lot of our patients are slightly older and find using Apple Watches a little difficult. The Fitbit is slightly easier. I'm definitely not a proponent for one or the other. We use those because it was the easiest, simplest model, but for sure. And I think as we move forward, these devices are going to get better at evaluating gait in a more quantitative way.
Do you find the quality of the Apple Watch data as good as Fitbit or better?
So the question was if I find the quality of Apple Watch data as good as Fitbit or better. And to be honest, I haven't used the Apple Watch for the studies yet, but it would be. There are a few studies comparing the different devices, and I think the Fitbit one which is actually one that you clip onto your shirt, came out better than all the rest of them, including Garmin. But that was now 6 years ago, so a new study should probably be done with the data.
Any surprises in terms of the reliability of patient-reported activity and kind of what actual activity people are engaged in?
So the question was, any surprises with patient-reported activity and the activity that's reported that we see in the. Yes, there was at the beginning of the Fitbit study, we looked at the MSWS12, which is a 12-item MS walking scale. So asked patient how they feel that MS has affected their walking, and by 6 months, because they were answering questionnaires every 3 months, there was a total disconnect between how they thought that MS was affecting their walking and their actual walking. And then once I think they'd seen the questionnaires, they sort of switched, and then it was more correlated towards the end of the year. So maybe it brings their disability more cognizance of.
Thank you.
Hi, thank you for allowing me to participate in this amazing symposium. My name is Roland Henry, in the radiology department, and today I'm going to talk about spinal cord imaging. Let me get my. Okay, and so today I'm going to be following up on the idea of transitioning to SPMS in relapsing MS patients and the recent identification of silent progression as a really important mechanism that's occurring in relapsing MS patients. In 2013, 2014, there was a consensus paper on MS categorizing a more improved categorization of relapsing and progressive MS. And these are the canonical features that go along with it, the conventional wisdom in MS, where you have relapsing MS that can be active, and worsening is, as you see highlighted here, is defined as increased disability following a relapse. And this paradigm has been greatly challenged recently with the occurrence of new work that's come out that shows that silent progression or disability worsening independent of relapse and MRI activity, also PIRA concept, is actually quite prevalent in relapsing MS patients, and especially in the new modern treatment paradigm where there are actually fewer relapses and MRI activity. And so the also longstanding concept of what is secondary progressive MS and how we diagnose it is also an issue. In this same paper from Lublin et al., they also point out that there are no clear clinical imaging, immunologic, or pathologic criteria to determine the transition point when relapsing converts to MS, and the transition is gradual. And I guess we would like to present that, in fact, perhaps MRI can play a role here, in particular spinal cord MRI. So silent progression, disability worsening in the absence of relapses, and MRI activity was shown very nicely from the paper from Cree et al. Using the UCSF cohort, and then Kapos also et al. Also showed similar results with PIRA, and in both these cases, brain atrophy was looked at and seemed to be also something that occurred even in disabilities worsening without MRI activity or relapses. But one of the things we wanted to understand were, well, what are the predictors really, if you look at demographic and imaging metrics, which of these could tell us about what's going on with relapsing patients who A, transition to SPMS, and B, have silent progression. We used the EPIC cohort of about 500 patients to conduct two experiments. One was to select 54 patients who transitioned to SPMS within 12 years of the study, and find a matching 54 patients at the beginning of the study who did not. And these are all relapsing patients. And then we could ask this question of which biomarkers and which actual metrics and characteristics predict which patients would have transitioned. So what's the difference? SPMS we used the most modern definition of SPMS diagnosis, especially based on the Larsheider paper, where they use the MS-based data and actually were able to diagnose SPMS some 3 years or more earlier than has been done conventionally in the clinic. And then we looked at this data in 2 ways, with mixed models to look at each of the MRI and the demographic predictors, and then looking at prediction of time to SPMS using survival models. Similarly, we looked at survival models to look at what are the predictors of silent progression in 306 relapsing patients we had in the cohort. In order to look at spinal cord area as one of the metrics, we have brain measurements that were done beginning with our cohort in 2004, but we were able to get the spinal cord measurement from the bottom of the brain image and solve some technical problems in order to make this a precise enough measurement to actually analyze the impact of spinal cord imaging. And this was work really led by our fellow Antje Bischoff at the time, who did a really remarkable job. So here are the cross-sectional results, looking at the different types of patients. We have controls in orange, patients in blue who did not transition, and the ones in red transition. And then we have data at the baseline on the left, and then there's a dot, and then they're separated by the data at the time of transition. And so what you can take from this is that at the point of transition, there's really not much of a difference in brain volume between these two groups. And then same thing with the white matter volume, maybe a little bit more. Gray matter volume, lesion load is a little higher in the group that transitioned. Ventricular volume looks very similar. Thalamus looks a little smaller in the group who transitioned, and cortical gray matter looks similar, while spinal cord area actually was significantly decreased. So that's the first indicator of what is the strongest predictor of this. And if we look at the overall results from the atrophy measurements, you can see that spinal cord area is the strongest predictor of the difference between the patients that transitioned versus those that didn't, with almost a 2 to 3 times faster spinal cord atrophy rate. And thalamic atrophy also trended in that direction, and you can see that there is a tinkling that percent brain volume change might also be important. If you look at the rates across this period of time, you can see the dramatic difference in the spinal cord atrophy change, percent change in the two groups. Here, the red group representing the group that transitioned to SPMS. And these are measurements that are occurring before the diagnosis of SPMS. If you look from a distribution point of view, here's a distribution of the cord atrophy rate group that transitioned, the red group again, versus a group that did not. And you can see that they're quite strongly separated after the analysis. And even in the group where the baseline EDSS was less than or equal to 2, there is still a very strong separation. So this fast atrophy rate is occurring in patients even when they have low levels of disability. In terms of the accelerated failure time model, this is the predicted time to SP diagnosis. And on the horizontal axis here, you can see the subjects with fastest atrophy rates actually have a predicted time to SP diagnosis of around 6 to 12 years. And that depends on their starting baseline EDSS. Of course, with the lower EDSS at baseline, having a little longer time to transition to SP compared to. So from this, even though we included all these brain metrics and demographic matrix, the only 2 significant predictors were cord atrophy, which was the strongest by far, and then baseline EDSS. And so for each 1% cord atrophy change, in someone, that predicts like a 53% shorter time to SP diagnosis. So we see that cord atrophy rate is the most important predictor of future SPMS diagnosis. it does it much earlier than the diagnosis. In the data set, we could support statistically at least 4 years before. In several subjects, in many subjects, it was even 10 years before they had this much faster atrophy rate of the cord. And so clearly there is a process that's underlying the transition to SPMS and the diagnosis of SPMS in relapsing patients. And this is present quite early in these patients and so far unrecognized. In terms of silent progression, we also looked at the prediction of time to silent progression using about 300 relapsing patients. And in that case, the significant predictors were, again, baseline EDSS. The strongest predictor was the cord atrophy. And from the brain metrics, the surviving predictor was the lateral ventricular volume enlargement. And you can see the different lines on this plot represent different levels of ventricular volume enlargement. It has a small effect, but you can see the tremendous impact of cord atrophy rate on the predicted time to a silent progression event, with the fastest atrophy subjects, the subjects with the fastest cord atrophy actually predicting about 2.5 years to a silent progression event. Each 1% faster cord atrophy was actually associated with 69% shorter time to silent progression. So what we've seen is this relapse-free and MRI activity independent progression. It seems to be present in the modern era in many patients, and in fact, almost all the worsening that we observed were of silent progression type or independent of relapses and MRI activity. We've seen that spinal cord atrophy seems by far the strongest predictor of both silent progression and SPMS. So the process underlying SPMS seems to occur very early in relapsing MS, and it's currently clinical unappreciated. And we can see it with MRI, spinal cord atrophy, and also brain atrophy to a lesser extent. This really, these data actually really challenge the way we think about relapsing MS. There's a nice discussion of this also in Nature by Philippi and Roca. I can refer to, but we have two choices essentially at this point. We either have to say that secondary progression should be diagnosed much earlier using other measures like atrophy metrics, for example, or we need to have a better or deeper way of characterizing what we call relapsing MS patients and their ability to get worsened. Is it time for spinal cord atrophy in clinical trials in the clinic? I would say yes. Treatments that impact spinal cord, would really be important because, those would mean that there's a really strong potential that these treatments could impact long-term disability worsening, silent progression, and transition to progressive MS. Clinically, again, characterizing patients early on who have these really fast atrophy rates is important. These patients need to be treated as if they're secondary progressive patients. And there are technical challenges, and I think that our group and many others are really working to increase the precision with which, and as you saw the presentation of the bridge paradigm, this is really where we need to be in the future, where we're bringing these metrics into the clinic and making them available to clinicians and patients so that they can make better informed treatment decisions and management. I would like to thank my group and the UCSF EPIC group, and in particular, Antje Bischoff and Nico Papinuto for their contributions to this work. Thank you. Are there questions? I'm not sure I'm hearing anything.
Roland, can you hear me?
No, they've muted.
Hello, Roland. Can you hear me?
Hi, I can hear now. Yes.
Ah, wonderful. That was a beautiful talk.
Roland, can I ask one question? Given the current technology, how many time points are needed to accurately project future neurodegeneration and secondary progressive MS?
I think we would probably need at least 2 to 3 years of data in order to do that. You know, the work is going very quickly, so I'm hoping that that will change in the near future. But assuming annual scans, I would say that at this point we would need at least 2 to 3 years of follow-up to establish the rate of atrophy. On an individual level.
Can you predict the future? How long will it take, do you think, to have a reliable, fully automated measure that could give us this critical data point for tracking our patients? And for the audience, I would emphasize that this is so important because we have treatments that are effective for secondary progressive multiple sclerosis, for silent progression, and for all types of secondary neurodegeneration in MS. So being able to track patients reliably and quickly is so important in therapeutic decision making.
I would say that we're already at the point where total brain atrophy could be used, or maybe ventricular volume. I think the precision there is sufficient. On the other hand, as we've seen from our study and even other recent studies where they've looked at the relationship of brain atrophy to silent progression, the effect size is smaller in the sense that it's not as predictive as spinal cord. So really, I think our aim should be to get spinal cord to that level. And I'm an optimistic person, as you know. And so I would say in the next 3 years or so, we should be at that level. Right now, I think, though, one way to think about it is that if we want to see the people who have the most extreme rates of atrophy, I think that will happen before we actually get the precision that we need to more subtly monitor everyone. So for example, if you have 3% atrophy rate, I think technically we'll be able to tell you that within a couple of years.
So Roland, at the Fondazione Prada event in September 2025, we will have fully automated spinal cord measures.
Absolutely.
Wonderful. Thank you.
Okay, thank you very much for the privilege of being able to participate in this great workshop today. So I'm going to focus a little on a different topic, which is antibody targets in multiple sclerosis. So as has been discussed earlier, MS is a inflammatory demyelinating disease that affects both the brain and spinal cord. And one of the hallmarks of kind of the diagnosing MS that's been observed for multiple decades now is that virtually all patients generate compartmentalized antibody species in the spinal fluid. And so it's a very useful diagnostic biomarker, which is also the case for other less common inflammatory CNS disorders. But one of the mysteries in the field about these ubiquitous intrathecal antibody species is what the antigenic targets are for these antibodies. We know the antigenic targets for much less common diseases than MS, but and we know lots about MS, but this has been a really difficult nut to crack. Antibodies are made by B cells. They're very mature forms of B cells. And there's no doubt that B cells play a critical role in MS pathophysiology, and that's really highlighted by this really stupendous clinical trial result by Dr. Hauser here, which showed that CD20 B cell depletion essentially eliminates relapses in patients with relapsing remitting MS. So there's no doubt that depletion of this particular subset of immune cell is critical to controlling the disease, but there's still confusion about exactly what role these B cells are playing in the pathophysiology of the disease. It's unclear if it's primarily the ability of B cells not necessarily to produce antibodies, but to present antigens to T cells and stimulate them, whether it's cytokine production by B cells or potentially autoantibody effects or other B cell functions that haven't been elucidated. And so a tool or a series of tools that we've been using for a number of years here to try and piece part of this out, especially the antigen specificity of B cells, has been to track clonally related B cells, so B cells that are likely to be specific for a common antigen in different body compartments, especially within the central nervous system. And so to do that, we take advantage of what's called the immune repertoire, and that's this recognition that both T and B cells recognize a huge diversity of foreign antigens in autoimmune disease, self-antigens. And this diversity is really, really remarkable. It's on the order of 10 to the 10, 10 to the 11th genetic diversity in terms of antigen ability to bind different antigens. And the sources of that diversity are also diverse, and they stem initially from just allelic diversity in the different immunoglobulin gene segments that comprise the B and T cell receptors. But then after antigen stimulation, B cells have the additional ability to undergo somatic hypermutation, affinity maturation, and class switch recombination to become further diversified and further able to specifically bind a particular antigen. We've, I think, all gotten an education about this in the last 2.5 years with the wide variety of COVID studies, both to immune response, both to the wild type virus, but also understanding how B cells evolve in response to mRNA vaccines. And so to track the immune repertoire in MS, so this work was initially done by Dr. Christian von Budigen when he was here at UCSF together with Dr. Hauser. And this was an early paper documenting kind of what we knew already, which was that, again, these oligoclonal bands or these immunoglobulin species that are persistent in the spinal fluid of patients can also be detected in another way by documenting with bulk immune repertoire sequencing that there are these persistent B cell clonally related cells that are expanded specifically in the central nervous system. And again, those can be tracked more specifically with immune repertoire sequencing the most genetically malleable parts of the immunoglobulin genes. And so this bulk sequencing is very powerful for kind of identifying these populations of cells. But at a bulk level, you can't resolve at a single cell level which heavy chain and which light chain in a particular B cell are paired, and then and so therefore you can't reconstruct that specific expanded antibody in the CNS, and it's more difficult to identify the antigen specificity. So a follow-up study that we published a couple of years ago took advantage of the explosion in single-cell technologies, both to look at the single-cell transcriptional phenotypes, but also the immune repertoire phenotype. And this work was led by Akshay Ramesh, Brian Schubert, Ravi Dandekar, and Ariel Greenfield. And in that study, we took treatment-naive MS patients and phenotyped single immune cells, both in the spinal fluid and in the blood. This gives a sense of the demographics of those patients. And for the first time, we were able to separate out at a single cell level kind of three large categories of B-cell subtypes, so naive, memory, and plasma cells in both body compartments, and could compare the phenotypes of these comparable cell types both in the spinal fluid and the blood. And thankfully, we were able to recapitulate what's been known in the sense that there's a large expansion of memory B cells and plasma cells in the CSF. And then we were able to look at the actual transcriptional phenotypes of those cells and found that indeed they were pro-inflammatory by many different measures, suggesting that, again, they really are involved in the inflammatory milieu in patients with MS. But again, now that we, again, have seen these cells in a different way, we still want to know specifically what antigens are these clonally expanded, highly inflammatory, compartmentalized cells actually going after. And so we've been, over a couple of years now, we've been using a number of tools to try and identify the antigen specificity of about 130 monoclonal antibodies that were generated from these cells. We've looked at their ability to bind rodent brain tissue. We've used a couple of programmable phage display tools that we've deployed here for a number of purposes at UCSF to identify across the whole human proteome and virome antigen specificity, and then also done more proteomic studies with immunoprecipitation mass spec. And these are just some examples. There's many antigens that we've identified thus far, and I'll just summarize those. But suffice it to say, one of the surprises we found across these 130 monoclonals is it's a rare monoclonal, specifically 2 out of 130 that are white matter specific. Indeed, many of the antigens that we've identified thus far by these various means actually don't target white matter proteins or myelin components, but rather are specific for a number of intracellular antigens that are very ubiquitous, not just in the brain, but throughout the body. And this just, again, is a short talk, but I'll just highlight a couple here just to show how we validated these. Once we get a candidate antigen by one or more of these assays, we then can validate it by cell-based overexpression assays. This is one example here, and then another one of a dynamic 2 overexpression assay validating that antigen for a particular antibody. So, this has been seen to a smaller scale in other studies that maybe multiple sclerosis is not a single antigen disease. And I think it, to the degree that many of these antigens are intracellular, raises the question as to how pathogenic the autoantibodies that we detect in CSF really are. And then I'll end with just highlighting another ongoing study that's been led by Colin Zemechnik, Gavin Soa, in collaboration with Dr. Hauser, Dr. Wallin at University of Maryland, Ravi Dandekar again, and Rebecca Baer. And this is leveraging this phenomenal biobank, the Department of Defense Serum Repository, where you can actually look back in time and acquire samples years before someone develops a disease of interest, in our case, MS. And we begin now in a polyclonal sense. We're looking at the serum autoantibody profiles using these same tools in patients years before they develop multiple sclerosis, and then years after. And the highlight thus far with the phage display technique is that we've identified a cluster of MS patients who who have a very specific autoantigen profile, on average about 5 years before diagnosis, and then that's been validated in a similar proportion of patients in the UCSF Origins cohort, which is a new onset MS cohort, and those antibodies are present both in CSF and blood. So I'll just end there and again just highlight that we and others have generated data suggesting that these cells that we already know are highly disease relevant are indeed inflammatory, clonally expanded in the spinal fluid, that many of their antigenic targets appear to be self-antigens that are intracellular, ubiquitously expressed, in that, at least in a subset of patients, it looks like there's a autoantibody profile that can pick people out many years before they actually are clinically diagnosed with MS. So I'll end there and just thank the many collaborators and funders and both here at UCSF and beyond. So thank you.
That was wonderful. Do you think that the oligoclonal band testing is 70 years old? What do you think the prospects are for a molecular diagnosis of MS based on oligoclonal B cells, either in the spinal fluid or peripheral blood? This would be a big advance.
Yeah, so I think the last study I highlighted, the Department of Defense.
Maybe worth repeating.
Oh, sorry. Oh, so sorry to repeat the question as to what would be a modern kind of equivalent for this oligoclonal band test that we do today when we're evaluating someone with suspected multiple sclerosis. And I think this last study I highlighted, Department of Defense study, gives me hope that there could be a more updated, more precise version of a diagnostic assay. And I think I've been impressed with that study that at least in this subcluster of patients that there's a very specific signature, again, that precedes clinical symptoms in many patients, and that those antibodies are very easily detectable, both not just in the spinal fluid, but also in the blood. So I think the question now, of course, is for those patients that lie outside that cluster who clinically look similar, can we find a companion profile? So it might be a family of diagnostic tests that are autoantibody targets that could help increase or decrease the likelihood that someone will go on to develop MS. So it makes me hopeful. Thank you.
Thank you.
Okay, so it's a great pleasure to be part of this exciting symposium, and hello everyone here and everywhere else in the world where you might be. So today what I will like to talk about, this is quite an ominous title, but I'll try to make justice to it by describing how are we trying to understand the gut microbes that live in and around us with the food that we eat, and how we try to put that all together informatically, because that's the only way we can do that. So some of you may be aware of the IMSMS. The IMSMS is a observational study that tries to understand the role of gut bacteria in multiple sclerosis. And the overall goal is to recruit 2,000 patients and 2,000 controls around the world, and understanding who's there. So by sequencing DNA of from stool samples from patients, and also what are they doing? So what is their function and how can we potentially intervene to modify this and restore normalcy? So this is the latest publication that just came out in Cell last week. And what we've done there is we've analyzed 576 pairs of cases and controls, and these pairs live in the same household. So they are most normally spouses or roommates, and this is the distribution. So almost 40% of the patients were untreated, and some of them were treated with the regular disease-modifying therapies. Most of the patients were like on the prevalence dictates, accelerometry. A few of them were experiencing progressive disease. And this these are some of the results. So when we look at the alpha diversity, so this is how many different species of bacteria can we find in the gut of these patients, we don't see a huge discrepancy between cases and controls. And that's expected. However, when we look at the beta diversity, that is the overall composition of the bacterial communities that live in patients and controls, we did find a statistically significant difference when we looked at all patients together, even when we look at all untreated patients together. And the bottom graph shows what are the other factors that might be influencing composition of the gut microbiota, and most notably is the recruitment site. So we're right in pairing our individuals within the same household because it's more likely they will eat a similar diet, which is one of the major components or modifiers of the gut microbiota. So this slide shows some of these bacterial species that we were able to find either more prevalent among MS cases, and those will be highlighted in red, or less prevalent among MS cases. And you can see there's many significant associations, and some of them will be associations only with relapsing-remitting cases. Some of them will be even associated with progressive disease, which is of extreme interest for following up. We also had the opportunity to bioinformatically look into what are the pathways that might be overrepresented by these bacteria. So what are the metabolic components that are being brought up by these bacteria that are present in excess among MS patients? And one of these pathways to call our attention was the phytate degradation pathway. And phytate is a plant-derived nutrient that gets metabolized by certain bacteria, including Akkermansia mucinifera, which has been the microbe that perhaps has been the most reliably associated with disease. And the interesting thing is that when phytate enters the degradation pathway by Akkermansia mucinifera, it converts into myoinositol. And interestingly, myoinositol is a metabolite that has been seen in excess through over time, both metabolically and through quantitative resonance imaging. So it is an interesting discovery. And the other thing that perhaps really caught our attention was that because we have several patients who were undergoing therapeutic treatment with different disease-modifying therapies, and our study involved a metabolomics component, we were able to see the metabolites both in blood and in stool as they were being modified by the different treatment modalities. So for example, we saw a peak on fumarate in serum in patients treated with a methylfumarate. So that was a good positive control. But we also found an interesting upregulation or a higher concentration in the serum of 3 key short-chain fatty acids. And short-chain fatty acids, namely butyrate, fumarate, there are and propionic acid, 2, 3, and 4 carbon short-chain fatty acids, they were increased. Their concentrations were increased in serum, but decreased in stool. What this indicated to us in individuals treated with interferon beta only. So this indicated to us that perhaps the transportation of this short-chain fatty acids from the gut to this, the main circulation was being enhanced by the drug. And in fact, when we looked at the expression of genes involved in these transports, which are required to transport actively short-chain fatty acids from stool or from the gut to the blood, these two genes that are key transport of short-chain fatty acids where the expression was increased in cells when treated with interferon beta. So this points a completely new potential mechanism of action for interferon, because we know that short-chain fatty acids are immunomodulatory. So if you increase their concentration in serum, they may have a potential beneficial effect. And lastly, the connection with diet. We know everything that the patients eat because we ask them through standardized questionnaires. And now we know that, for example, MS patients eat, in general, a healthier diet. And this is perhaps after a diagnosis, they try to take control of their disease by eating a healthy diet, which is always a recommendable step to take. And here you can see that they eat more fruits, more vegetables, more refined grains, and also a healthier variety of fatty acids. And we are now starting to be able to correlate individual microbes with individual nutrients. So now we know if we want to increase the presence of a particular microbe, now we know which is the nutrient that it might be linked to, and we can modify or suggest a modification of diets in order to increase that. So this is just starting to happen now. And how to put that all together? Well, for that, we need a lot of people who are super intelligent, super bright in 20 different fields of biomedicine, or we can use computers. And we decided to use computers in addition to very smart people because computers have faster processing time, they always remember things, and they can put things together at a scale that humans cannot. So this is, Dr. Bove mentioned briefly, the spoke knowledge graph, which is essentially the database of databases. So the spoke knowledge graph contains more than 45 different databases that we, as scientists, consult depending on our specialty at different times. But this is all the information together. More than 50 million concepts related by more than 100 million relationships with semantic value. So they're biologically meaningful relationships. And based on this, we can start making connections. So for example, this is the entire bacterial taxonomy of more than 5,000 bacterial species that are found in the human gut. So we can navigate up and down, zoom in and out through a visualization system, all the bacterial taxonomy of bacteria that are important for human health. We can also look at metabolic reactions. I mentioned metabolism. So we can zoom in and out and explore any metabolic pathway that we want. In this case, central carbohydrate metabolism is blown up. And then if you focus in on one of those reactions, then you have the Mayer-Foch pathway, which is glycolysis. And you can see how the glycolysis produces pyruvate starting from glucose, and you get every single enzymatic step depicted in this, and you can choose how to branch off of that. You want to know more about what else contains glucose, then you blow up that glucose node. You want to know where this pyruvate goes after this, well, then you can blow up that node. And other questions that we can ask through this is, I mentioned the importance of butyrate, for example. Can you tell me what are all the bacterial species that live in the gut that have the enzymatic machinery to produce butyrate? Well, there you go. So how about serotonin? So we know that 85 to 90% of all serotonin is produced in the gut. There's bacteria that carry that machinery, and we can identify them through their encoded enzymatic with the encoded proteins with enzymatic activity. Likewise, we can identify all the gram-negative bacteria because they produce LPS, and which ones are those? We can do that very, very effectively and very quickly. So just to close, I would like to leave you with an example with, again, as food for thought, But if you type in multiple sclerosis in Spoke, and then you get this type of relationships towards the left. All of those are diseases that are somewhat similar in the literature too, because they share certain characteristics. Towards the bottom, you see the subtypes of multiple sclerosis, and towards the top, you see what kind of disease multiple sclerosis is. So this follows from the top to the bottom, follows the disease ontology, and on the left-hand side, you have diseases that may resemble MS. So now you can, as I mentioned, you can blow up this graph in any way you want. So you can say, okay, now I want to know more about, for example, the symptoms of multiple sclerosis. So you blow up and then you get some of the symptoms in pink. And if you want to know what are the genes associated with multiple sclerosis, you can request this, and immediately you get all genetic associations with MS. You can decide to go, okay, these genes encode which proteins. You can go that route. Or you can say, I want to know all the drugs that are being used to treat MS. And there you go. So these are all the therapeutic approaches that some of them are approved, some of them are in development. You can choose whether you want to do phase 1, phase 2, phase 3, or approved. And just to close, how we are trying to integrate the bacterial population analysis that we're doing through the microbiome project with food intake and again, the physiopathology of multiple sclerosis. Here you get the entire taxonomy of Akkermansia mucinifera, which is one of the organisms that has been associated the most with multiple sclerosis. You can go all the way up into the kingdom of bacteria, then phylum, class, order, genus, and species. And then if you blow up Akkermansia mucinifera to tell you what are all the proteins that this particular bacteria encodes, you get all those in green. And some of those proteins, like in bacteria, usually 1/3 of the genome is devoted to enzymatic potential. So all of those proteins that are enzymes, you will get another note in orange telling you this is an enzyme and this is the catalytic activity that this enzyme is associated with. And then exploring this, I found that one of those enzymatic, those catalytic activities is this one, that in fact, what it does is it produces a metabolite called biotin. So Akkermansia mucinifera has the ability to produce biotin from dietary input. And of course, biotin, there's been clinical trials linking biotin and multiple sclerosis. They've not been extremely successful, but we still know the no. And this is, again, provides some reasonable chain of thought and based on evidence. In each of these steps, you can gather where the information comes from. And one last thing that we, just to close the loop, we know that biotin is present in several foods. So again, this is all contained in Spoke, and you can see that, for example, the pearl oyster mushroom contains a lot of biotin. So again, you can see very, very quickly, you can go from molecular to pharmacological to food to lifestyle with this wonderful tool. So I'll stop here thanking everyone who participated in this from my lab, the MS Center, and all our collaborators in other institutions. Thank you so much.
Sergio, thank you. You mentioned bacteria are doing so much through their metabolites in influencing homeostasis. You mentioned the importance of serotonin synthesis by bacteria. Given Dr. Bove's comment about the importance of monitoring mood, I would think that that would be a relatively easy question to ask, if there is an association between mood and a particular species of bacteria.
Yeah, let me repeat the question. Dr. Hauser asks, how can we, or whether we can link the information that we've gathered from this cohort of patients on serotonin with other projects that were described earlier that assess mood in MS patients. And I think that's a terrific idea. We definitely have serotonin levels measured by metabolomics in almost 500 patients. So we can definitely try an association. That's a great idea.
Yes. Sorry, have any of those been linked with fatigue? Because that's something that's really difficult to treat and diagnose.
Yeah, so the question is, what about fatigue? And in our questionnaires, we do include a question for symptoms, and I think fatigue is one of them. So it's hard to measure, and we're not experts in determining the different levels of fatigue that an MS patient might be experiencing. So we may have a semi-quantitative at best, but this might be something to look at. Yes.
Yeah. So how about, as you mentioned that the biotin is somewhat very interesting through that software which was used. So is there any study or anything known about biotin deficiency which is related to associated with defects in B cell immunity? So that could be interesting because we know that the CD20 depletion therapy is quite effective. If biotin has anything to do with the defects in B cell immunity.
Yeah, the question is there anything now about biotin and B cell immunity? Honestly, I haven't made that link. I think that, again, with something like this, you can play, and in fact, there's cell type nodes here that are linked by the proteins that they express and the genes that they express. So one could potentially navigate from biotin to the cells that produce the enzyme that produces biotin to the cell types that express that gene. But also, of course, talking to experts in B cell biology would be a good idea, but I'm not aware.
Thank you.
Thank you.
Well, we will now set up for a tableless roundtable discussion. So I might ask all the speakers to join. I don't know how it is at other centers at UCSF. Masks are strongly encouraged. So should we keep masks? Why don't we do that?
I'm supposed to be okay.
Just about 17 texts.
Yeah.
Your speakers. Dr. Hauser.
I don't need it. So maybe I will start it. I think throughout, maybe we could take our mask off when we speak. Dr. Wilson showed the clinical data of high efficacy B cell therapy in multiple sclerosis, which, as was shown, has spectacular results against inflammation that we can measure in the brain through an abnormal blood-brain barrier and through clinical relapses. But unfortunately, only partial benefits against progression, which is the key topic of this morning's conference. Progression in MS, as Dr. Henry showed us, is related, we believe, to chronic inflammation trapped behind a closed blood-brain barrier, making our therapies impermeable to the site where they are needed. And the holy grail in multiple sclerosis research, an area that has had enormous progress over the past generation, is to more effectively target this inflammation driving progressive loss of function, which we believe is neurodegenerative. So my question is, given that the landscape of therapeutics under development are mostly targeting this great unmet need, MS progression, which monitoring tools do you think are ready for prime time in this arena? And Michael, maybe I would begin with you. As you know, there is a trial called IMPACT, led by Dr. Bruce Cree at UCSF, that is attempting to use high efficacy B-cell therapy at the very onset of disease to extract greater benefits against progression. And oligoclonal bands are one of the key primary outcome measures. So I might begin there. Are there immune outcome measures that you think could be useful surrogates against progression to give us an early signal?
I think that to the extent that we believe that the progression is at least in part due to this stubborn inflammation that persists in the CSF or in central nervous system as a whole, despite high efficacy therapy. I think that early treatment with B-cell depleting therapy, to the extent that it can better deplete those B cells that we know remain resident in the central nervous system, I think that that's an exciting strategy. In terms of monitoring, I think, crudely seeing the disappearance of those oligoclonal bands, we hope is one surrogate marker to suggest that that patients will be at a lower risk for developing progressive disease. We'd like to, and we know there's other blood-based biomarkers like neurofilament light chain levels that can be monitored more easily, that could indicate that patients are suffering less neurodegeneration over time. So I think that certainly, again, to the extent that it's those CNS, persistent CNS resident B cells that are culprits, I think antibody-based measurements, whether those antibodies themselves are pathogenic, could be great biomarkers to monitor whether we've achieved kind of what we're hoping to achieve in those therapies.
Dr. Henry, we will run the risk of this not working. Dr. Henry is quarantined for a few more days. Roland, can you hear me?
Yes.
What MRI measures of neurodegeneration are ready for a prime time for progressive MS trials?
I think that at this point, in the context of progressive MS trials, I think spinal cord and brain and regional brain metrics are all viable. The cohort advantage gives you the statistical power you need to actually get a treatment effect. So I would say a properly powered phase 3 trial could be done with spinal cord, brain, thalamus, cortical brain, all of these things.
Do you think that the spinal cord imaging will require dedicated spinal cord scanners, or can you get those from the routine brain scans, given that you're high in the cervical cord?
We're really on the precipice of understanding that a little bit better. If you use modern high-resolution brain images, you can do pretty well, but you will always get better precision by doing spinal cord dedicated scans. Now, these scans don't necessarily, you can just monitor the upper cervical cord, and therefore you actually don't need any special arrangement on the scanner to do these scans. You could just use the same head coil that you use for brain imaging and so on. So it's really not a major interruption to add in an upper cervical cord scan to a clinical trial.
Don't know if others want to weigh in on this question about how we should monitor progressive MS trials. Dr. Block?
I think as Dr. Baranzini had said with the biotin trial, sadly the trial didn't meet its endpoints, but we were able to show it was feasible to measure physical activity in 600 people, 90 different sites, 10 different countries. And we've shown the data I showed before that we can see change sometimes before we can see change in clinical metrics. I think it will be a really important part of trials going forward.
I might shift gears for now and move to a different area, and a question initially to Dr. Bove. One of the great challenges in displaying large amounts of data is the risk of incidental findings, which are defined as findings that we don't expect, we're not looking for. And chasing after incidental findings is a potential source of increasing the cost of medical care, and also many iatrogenic complications follow invasive procedures that follow the identification of an incidental finding. So how do we navigate that terrain of the certainty of incidental findings appear when so much data is presented to patients and their physicians?
Excellent question. I think for the purposes of our disease-specific dashboards, where we're really pruning the data, really selecting, curating data for visual display, that problem is less because we sort of figured out which are the things that we really want to focus on and see. That said, as we pull in data from Spoke, from these enormous knowledge networks, that's going to really have to be a part of the roadmap in terms of clinical validation, clinical utility. Are we getting sort of more information than we otherwise, or are we just getting saturated with incidental illness that are actually distracting from the goals of monitoring how our patients are doing? I think we can't really, we've never really had that degree of data available at the point of care. And so part of the roadmap and the user engagement and the process of implementing these types of tools is to figure these kinds of questions out. Because we've never had it in the hands of clinicians before, and that's what's actually really exciting.
Yeah, so in my view, if we're really serious about preventative medicine for the next to be implemented for real in the next 20 to 30 years, I think we'll have to start dealing with those issues from the legal standpoint now, because incidental findings are only incidental because the specialist who's treating the patient is not looking for them, but they still pertain to the health of the individual. So they're not incidental for the individual. They could be potentially life-saving. But I think that when we're looking at now, as always, technology goes ahead of regulations. And with technology, now we can sequence the entire genome of an individual and find many incidental, so-called incidental findings. Some of them with now regulations say, okay, yeah, this is actionable now. And I think there's going to be more and more of those examples coming up from integrating and cross-referencing information from multiple data streams and modalities that someone will have to care about in order for the patient to be better served. So I think that is a very, very profound question that we'll have to deal with shortly on the terms.
Others in the audience or on Global Network who want to weigh in are welcome to do so. Please just raise your hands. Maybe I will ask a question to Dr. Block related to your presentation. You spoke about the EDSS, which is our mnemonic for the Extended Disability Status Score. It's the validated composite score of disability that we use to monitor our patients and grade our patients over time. It's a misnomer. As you know better than I, it's not a disability score, it's an impairment score. It's measuring mostly what we detect on our exam. It's not telling us if a patient can dance with his wife, if he's enjoying life, if his slight changes in his finger function is impairing his work ability. So how patients care about their impairments, the things they can't do. We measure, patients care about their disability, the things they can't do. We measure impairments. How can we move towards a more patient-centric measure of disability as we're monitoring?
That's a great question. I've been thinking about this a lot as I move from monitoring physical activity and then bringing in another symptom, bladder. And incontinence was something I noticed a lot in patients who were physically able, their EDSS was low, but they weren't doing a lot of exercise. And so as I saw these patients fairly often, a lot of them were not doing exercise because of their bladder impairments. And that wasn't necessarily captured within the EDSS. So I think combining our clinic measures, which are obviously important, and so far that's what we have, and those studies use the EDSS as an endpoint. Combining that with our patient-reported measures and then remote measures as well to see if what they think they're able to do is actually what they're doing in the clinic, or whether they're telling that to us as clinicians because they want us to feel like they're doing a lot. So measuring continuously, we can see not just a snapshot of function, and maybe not even just what they are at baseline, but over years at time, how they're progressing.
Just to add to that, I think it's helpful to think about this, the discrepancy in multiple sclerosis is a very strong discrepancy between what we do in the clinic and how we grade all of this research. And in the clinic, when we're working with, we're very clear that there are visible symptoms and invisible symptoms. And we're also very clear that managing the disease state, active or not, progressive or not, is one component of MS care, but managing the symptoms, managing the comorbidities, and then thinking about the person, how they're doing in their life and setting them up to thrive. That's all part of our clinical care. When we switch to the research world, we've really been stuck on that first piece, are they active or not, progressive or not, and we forget all those other things. And so we then come to this term of silent progression, which is not silent at all. It's just we're deaf, right? Our patients are telling us that things are worse. They're telling us about their function in the daily life, and we are grading them as stable because we haven't yet seen our hallmarks of progression. And that huge gulf between the patient experience and what we can see with our limited tools is, I think, what we're really trying to bridge with all of these approaches, and the patient voice, the patient reported outcomes, and how they're functioning is all part of bridging that big divide and helping us to identify change sooner and prevent change sooner.
Dr. Block, I think another example of this is what you taught me just last week about how patients actually use ambulation aids and why our EDSS that separates cane from bilateral support may not be useful to patients. And maybe that's worth telling the audience about what you've learned about cane dependency, at least in people with multiple sclerosis.
I'm not sure
So that people are not using their canes. They don't find that helpful, and they're actually
right.
using either no support or bilateral support.
Right. So the need for a cane doesn't always translate into patients being more functional or physically active outside the clinic. So using the cane properly, using the right type of assistive device. I was helping someone up the stairs as they held their cane, and I helped them walk up the stairs. And she was a patient of mine, so I wasn't sure I could intervene. But you're right. It's there's a big discrepancy between those, and I think that's where physical therapy as well can come in and help educate and train patients to use those devices.
While we're discussing these monitoring tools, Dr. Bove, I might ask you to just say a couple of words about your diagnostic corridor that was just launched in the building next to where we're sitting today.
One of their goals to try to sort of increase granularity and sensitivity to change is to sort of digitally phenotype patients and to come up with this sort of a digital EDSS that's sort of a little bit more sensitive, more granular, more objective, and sort of less limited by the focus on ambulation or on sort of cane dependence as a form of ambulation that the EDSS is. And so we have launched sort of a neuro corridor or brainwalk that we're calling it, where we're able to capture gait metrics and many types of gait metrics on a carpet, and then also capture cognition, voice, and many other measures on sort of using tablet-based assessment following the walking, and together come up with a sort of multimodal score of function that's again more granular and visualized directly here. So we've been able to receive funding for this and to launch this. So it's a very large collaboration actually across all of our Weill clinics, so that we're able to capture data on patients from different neurological conditions, and that'll also help us be able to more sensitively and earlier figure out which patients should go to MS and which patients should go to movement disorders and the other clinics. So we're hopeful that we'll generate lots of multimodal data as well in the years.
Maybe as a last question, I will go back to something that I think Sergio mentioned, but we passed over. The questions of bioethics and constraints on bringing metrics that might not be formally validated to the clinic in a real-time manner, whether or not they're acted upon. So this is such a difficult situation because we are making judgments in the clinic every day and making an interpretation, Roland, from an MRI scan that the atrophy has worsened, and yet that is not a formal quantitative measure that we use. So, Dr. Bove, in terms of sharing data, or Dr. Wilson or Baranzini or Block or Henry, how should we think about sharing with our patients and even with the treating physician these research data that seem so germane to the patient's problem?
I think the first thing is we just have to be very clear that what we're doing is clinical research, and the patients have to be formally engaged in research and consented for research. And And then we have to explain, and clinicians as well. And we have to be very clear that at the end of the day, the clinician has many, many types of data to interpret. Clinicians are interpreting, we've always relied on the neurological examination as a form of information. And that information may be incorrect or inaccurate or partial. And so, yet, as clinicians, we have to sort of parse out what's known and unknown, what we trust in that data point, and figure out how to communicate that to the patient. And I think it's the same with all of these other data points. They're partial. They need to undergo rigorous validation in the sort of research world where we're looking at, backwards. And then the step of actually doing prospective validation to see if it actually matters has to be done absolutely thoughtfully and engaging patients in the process and engaging clinicians. And at the end of the day, they're the ones who have to interpret the information best. And that's what clinicians do. And that's what sort of clinical, exercise, medical training actually teaches us how to select data points that we trust and make our most informed decisions in uncertainty. And I think that's just an extension of what we're asking clinicians to do.
Perhaps
Thank you. Dr. Henry, I was going to ask you to weigh in because MRI seems so much a part of this today.
No, absolutely. I think that we need to be super careful here, and we need to be able to really not only provide metrics, but provide confidence on the data that we present. That's just on the one hand. On the other hand, as Riley was saying, we really need to have strong validation in the research cohorts, and there are different types of patients, and we shouldn't just assume that something that we see in a specific cohort or one type of patient is valid for all patients as well. The last part I would like to say is that in terms of sharing data with clinicians, I think that clinicians are used to integrating various types of information to make management decisions. And we should probably think about formally doing the kind of work where we can integrate information. And I think Sergio mentioned this before, where we actually do the validation, not just on singular metrics, but on the integrated information that a clinician might see in order to say whether it's actually useful or not.
Thanks, Roland. Well, we are at the top of the hour. I'd like to thank the 5 members of the team this morning for their fantastic presentations. Special thanks to Dr. John Van Leeuwen, without whose organization and assistance this would never have happened. Thanks to Shannon Warto and Andrew Barnegat for making sure that this would work despite the COVID pandemic. And thanks also to the Fondazione Prada, to my friend, Dr. Comey, and to all of you who have joined us. We will be meeting in person next week in Milan, and I'm sure that is going to be a spectacular event. So thank you for honoring UC San Francisco as one of the sites for this global initiative, and goodbye.